Okay, if we could have your attention, please, everyone. It's my great pleasure to welcome today Maria Briones to give this plenary lecture from pots to large-scale monitoring. Maria, I'm sure, is known to many of you. She's Professor of Soil Zoology at the University of Vigo in Spain, where she's been for the last 30 years, as she just told me. And every summer, or most summers, she comes to the north of England for the improved climatic conditions to work at the UK Centre for Ecology and Hydrology. And really, that's next door to where I work, so Maria and myself are in pretty much the same institution, so we see each other most summers when she's visiting us up in the north of England. Maria has a long-standing involvement with the GSBI. She contributed significantly to the Global Soil Biodiversity Atlas, and she also contributed to the Global Soil Biodiversity Assessment. So she's a long-serving friend of GSBI and hopefully will continue to contribute to the GSBI in the future. I think when I first met Maria, or became certainly aware of her work, was when she was actually working up in the north of England, very close to where I was also studying at the time, doing my PhD. And at the time, she was working on enkitrade worms in peatland soils. We have a lot of deep peat, cold, wet conditioned soils. But what was particularly innovative about Maria's work, that she was doing some, I would say, some of the first truly functional work on looking at how soil animals respond to climate change and the consequences for things like carbon cycle feedbacks. And worms, and enkitrade worms have been, she just described herself to me as a worm lover. They've been central to her research ever since she first began. But recently she's become more interested in broader issues related to soil biodiversity and the integration of that knowledge into policy. So I won't go on too much longer. I'll pass over to Maria, who's going to give us her presentation. So if you'd like to welcome Maria to the stage. Thank you.
Thank you, Richard. I think you can give my talk now. So I don't need to present the title. I will just go into the topic. So first of all, we know that soils are degraded across the globe. 33% are already degraded, and we are on the way of more or less degrade everything that exists on the planet. In Europe, the figure is around 60 to 70%, and that costs a lot of money to restore those soils because of the ecosystem services they provide. So that is why the political are centered now into recover these soils and implement sustainable practices. If we talk about soil biodiversity, then there are, according to FAO, these are the 10 threats to soil biodiversity. And I was lucky enough to participate in this report for opportunities for soils in Europe with Wim van der Putten and some of the other colleagues. And we ranked 5 big threats to soil biodiversity causing losses, which I could just make in 2 big drivers, which are climate change and land use changes. Unfortunately, despite we all know that soil biodiversity protection is never included in those policies that address climate change and land use changes. So now we come to the European Soil Monitoring and Resilience Directive. I was happy to be involved in the FAO-sponsored launch where we're discussing all this. So this directive was proposed in 2023, and then it was open to the member states to produce critiques, and I was in one of the groups criticizing this, but it has good things. At least we have a monitoring and resilience directive, and for the first time, it provides a definition of soil health, which rests in 3 pillars: the physical, the chemical, and the biological good conditions. So the biological, for me, is the great thing to include. It's for the first time. Before, soil health was based on chemical fertility and all these type of things. So, and then it came into force in December last year, and according to the directive, member states have 3 years to implement, to suggest a monitoring program, and they have to measure some biological descriptors, which are indicated. I was hoping I have a pointer here, but no. Do I have a pointer? No. Okay. And these are the descriptors. There is one which is compulsory, which is the metal barcoding. This is just because it's in Lucas. Of course, the ERC are behind this supporting this directive, so of course it's better if we have a comparison. And then all of these, all the rest are optional. And that is part of the amendments. I managed to, in my group, to put earthworms in there. But I was wondering what happened to enkitrace, isopodes, mites. There's nothing there. So that's why I'm going to now move in showing which are the main players in the different soils. In collaboration with Augusto Zanella and Ponch, we produced this classification of humus forms and which were the main actors. And as you can see here, in the more soils are mainly bacteria and fungi, but also enkitrace are playing a role there. But if you go to the mother and mule soils, it's mostly worms, which is one here. So I will start talking about the more soils. The peatlands are the most threatened habitats in Western Europe. They cover approximately 15 million hectares, represent 12% of the global peatlands, and approximately half of them are degraded. This makes Europe the second CO2 emitter, and also historically the largest emitter of CO2. Some of them, at least 20%, are incorporated in protected areas, if that means something, and they contain several habitats, according to the EU, and 3 of them are endangered, and 1 is critically endangered. So peatlands have been in my heart for a long, long time, as Richard said. So this is one of the places I've been working a lot in the UK when I was visiting, which is the Moor House. I think the name is quite right to the point because it's more. And the conditions in there, which is makes peatlands so characteristic, is the low temperatures, the low pH, and the waterlogging conditions. And that restricts biology. So there's only a few guys that can live in these conditions, which are tardigrades, mites, collembola, and also enkitrace and nematodes. So usually small animals. There's no earthworms here. When you look at the biomass, what the people say, even if enkitrace and nematodes are much bigger in numbers and density, when you look at the biomass, enkitrace wing are the champions in biomass in these systems, even bigger than the sheep. And believe me, in England, there are too many sheep on the grasslands. So that's why I started to work on enkitrace. They have to be important in this system. So my first question was, how do they respond to climate change? So I went to that Moor House, to the hill, and I collected soils from the top of the mountain, and I moved them down to in another location in the same slope, the same orientation, but warmer. And what I saw is when I have warm conditions, I have more enkitrace. So they like it warm. Maybe because they are British, I don't know. And then the other thing I noticed is when in the summer, when the surface was getting really dry, they were able to move down. The literature was saying that they were concentrated mainly in the top 3 centimeters, but actually they can move down to 10 centimeters. And then my next question was, what do they eat? Because usually ecologists say, well, organisms occupy a layer and they are directed on the process that are occurring in that layer. But what I found is it doesn't matter where they are, they always go to eat in the same place. So they always consume organic matter that is not really very fresh, it's at least 5 to 10 years old. So the next thing is, well, I have more Enki trace when it's warmer. What happened to the CO2 emissions, to the carbon exports? So I went back to the lab, to pots, and I started incubating soils with and without Enki trace. And when I didn't have Enki trace, and at 4, 10, and 15 temperatures, more or less I didn't get any difference in CO2 emissions. But when I have Enki trace, especially in the warmer temperatures, I have more CO2. And when I calculate the Q10, which is something that the modelers like to use, I saw that most of the models use Q10 equal 2, but when you have Enki trace, you have more than 3. So the next thing, my next question, this talk is going to be about questions, all the questions I pose to myself. So are these Enki trace playing a similar role in other ecosystems, in other peatlands? So I sample other places across the UK, Ireland, also include my Spanish peatlands and tutorial soils. And what I found is that, yes, if you took at the whole range of soils, the ratios between carbon and nitrogen, carbon and phosphorus, as long as you have high carbon, you are accumulating carbon in that system. But in the boreal soils, the main players were fungal microbes. So the relation between, especially fungi, were the ones going for accumulating the soil, the carbon. But in peatlands, it was Enki trace and bacteria. So depending on the system, you have different key players. So my next question was, what about other carbon exports? I was interested because I heard there was a Nature paper saying that there was a lot of soluble carbon in the soil solution exporting from peatlands. That was in Nature. Then I heard my colleagues that they were looking in the same system, DOC, and they were finding more DOC, but they couldn't link it to any factor. I mean, they were thinking, well, plant productivity, maybe phenolic compounds from the vegetation. So I thought, why not Enki trace? So I started again incubating soils, different layers with and without Enki trace. And then I collected the leachates, and I saw when I didn't have Enki trace, the top layer got the DOC, most of the DOC, because it's where the plant roots are. But then when I have Enki trace, because they move up and down, then I have more DOC in all the layers. So we repeated this experiment with one of my PhD students back in Spain to understand the mechanism of why it was more DOC into the soil solution. And what we found was that, you can see this is without Enki trace, and the other two graphs are with Enki trace. And the top graph is the pH. If I didn't have Enki trace, the soil solution was becoming more acid, whereas Enki trace were buffering that solution. And the other thing that happens is that when I decrease the acidity of the soil solution, there is more iron release. And with iron was all the organic matter also leached through the system. So they also play another important role in carbon exports. So my next question was, if I have Enki trace in our system, I know what they're feeding on in the field, do they change the feeding if I force them? So again, in pots, I started cultivating intact soils with Enki trace at different temperatures. And what I saw is when I have more Enki trace, they feed on much older carbon. And also, I was able to look that there was a tipping point when I was overpassing more than 2 grams per square meter, all the carbon sources were assimilated by the Enki trace. So I call it this feeding flexibility. So the animals feed normally in certain carbon sources, but if there is the system is stressed because it's warm, then it will feed in other things. So then my next question was, well, if I force the Enki trace to eat in assimilate all the carbon sources, doesn't mean that the carbon that is also export from that system is also older. So I did another incubation experiment, this 15 and 20 degrees, and I split the soil layers. So I was forcing my Enki trace in the deeper layer to feed, or to die, to feed on older organic matter. And then I look at the export carbon export, and I found that that was true. I have more Enki trace in the top layer, as you can see, but in the deeper layer is where I forced them to eating older carbon sources. That is where the DOC was older. So it was moving carbon sources that were in that peatland stored for a long, long time that become mobilized. So then I thought, well, I did all these experiments in pots about CO2, DOC. Can I extrapolate this really to the field? Do I know how the system works? So I went back to the system. And I moved again course, but this time I left them in the field for 13 years. And that's why I keep coming back. And every summer I was taking samples and measuring. And what, to our surprise, was all the CO2 was emitted from the warmest site was autotrophic. There was no differences. There was no release of older carbon sources. And actually, when we partitioning this CO2, 2/3 were coming from plants, from roots. So then I look at the DOC, and that was a bit different story, but contrary to my expectations, the older DOC came from the cooler place, not from the warmer place. And I thought, well, that's intriguing. So what happened to the Enki trace? And the Enki trace more or less make a switch. They started feeding on recent sources, but then towards the end, in the cold site, they started to feed on older sources. So how do I interpret this? Well, because in my pots there were forced and there were short incubations, as long as they have material to eat, they will eat what is available. But in the field, I have several seasons. The cold site restricts plant growth, plant exudates. The only very few windows when they can grow, because that area, as you saw in the first picture, is covered by snow. So that means there's not many fresh labile materials. So that's why in the long term, the animals don't have enough labile material to eat. Whereas in the warmer side, the plants are always replenishing the carbon. And that's why the different response is based on the same mechanism, but the interpretation is different, completely different. So my next question was, well, I know a lot about peatlands in the UK, some on my own region. This is my region. That is where the peatlands are. But actually, peatlands are complex ecosystems. I consider them as mosaics. So we have different habitats in the EU. The Habitats Directive considers several classifications. They put numbers to them. So my next question is, should we design monitoring programs based on the habitats, the plant communities, because the Habitats Directive is based on above ground plant communities. So I target some of these priority habitats, and I went back to this time to the field, and I selected 4 of these high priority according to the EU. And this represents a gradient of moisture going from the dry with the heather to the most moist habitat with the sphagnums and different quality. So the first question was, I could link soil biodiversity community structure to different vegetation types. Well, yes and no. With the mosses, it was very clear that there was, most of the time, they were dominated by diptera larvae and Enki trace because it's a wet environment. But with the vascular plants, I was finding a greater dominance of mites. And especially during the summer months when the heather was really dry, the mites were completely dominating the community. So that brought me to the question, it's very important when you sample, because even if you sample the shrubs in January, you get a completely different community as if you sample in July. So just getting one sample, it doesn't tell you about the community of that place or that habitat. So what we found is when we look at the whole data set, is we found that there is a gradient of moisture that leads to communities becoming dominated by Enki trace. When we have vascular dominance, in dry conditions, we have a community that becomes dominated by mites. And with that, we also get more CO2 emissions. So this is important because there was a lot of papers coming out in the literature saying that peatlands were threatened by vascular dominance, by shrubs. It is what we would call a shrub encroachment. So my next question was to find out how this vascular dominance was affecting the communities. So I went back to the field, and this time I wanted to know whether it was more important the moisture or it was more important the substrate quality. And I do that by collecting cores and half of them turn it upside down, because in that way I reverse the substrate quality gradient by expose the deeper layers to the same abiotic conditions as the top layers. And I did that in two habitats, the most extreme ones in terms of moisture. We collected enky trays, we identified several species. I only show here the general graphs, but we have individual responses. And most of the enky trays were stayers. What that means? Well, it means you look at the top half of the graph, that is the vertical distribution of 3 layers. So the top is at above, and then you have the middle layer, and then the bottom layer. And the second half of the graph, of each graph, is the opposite. So now the top layer is at the bottom, and the upper layer is what was the bottom one. So it means that in most of the times, the animals, even if you turn it around, especially in the shrubs, they were staying there. They were not going up to be exposed to the wet surface conditions. So to me, that responds that substrate quality is the most important thing driving the vertical distribution. And it doesn't matter if you have a shrub or a moss. We saw the same response in both of them. I also measure the carbon dating of the enky trays, and when I have more enky trays, they also eat older carbon sources. So it was repeating again the same things I have seen in the lab. So I wanted to complicate this a bit more, and then I just focus on vascular plants, and I wanted to know not only what happens to the enky trays, what also happened to the processes. So this time I took bigger blocks on 3 habitats, and I did the same thing. I turned it upside down. I look at microbial biomass, I look at enky trays, and more or less I got the same thing. The shrubs have a distinct vertical distribution more than the other vascular plants, but the animals tend to stay in the top layers. It doesn't matter if you turn it around. But when I look at the carbon spore, the CO2, it didn't care about the substrate quality. Here, out of the 3, the best for this composition is the middle one. It's a grass, it's very palatable, it's rich in nitrogen, so it's very easy, attractive for the decomposers. But what I found that for the CO2, temperature was the driver. So when I have a drier system with warmer temperatures, I have more CO2. Of course, I don't have, if I turn it upside down, I don't have many labile compounds, and that's why the CO2 emissions are less. Whereas for the DOC, it was substrate quality, what was important. So in the Molinia, which is the middle graph with the greens, most of the DOC came from the top layer, even when I was turning upside down, actually concise with the layer where my enky trays were there massively. So this is about enky trays, and this is about a pristine ecosystem. So I hope I convinced you that when we look at peatlands, more soils, we have to look at enky trays as indicators. But when we look at other systems, like mull soils that most of them are converted to agriculture because they are good to grow food, then we have earthworms. So I was in a soil mission week, and I heard somebody saying, why should we measure earthworms? I mean, they're everywhere. They are not good indicators. So I want you to convince otherwise. And it might be true in some cases. For example, in this paper, that we did in the Soil Agro project, we didn't find differences between management systems, conventional and organic for earthworms. But when we compare all the literature available with Olaf Smith, we did this meta-analysis, and here you can see on this left side the comparison against conventional treatments, conventional tillage, sorry. So the first one, reduced tillage, then deep soil loosening, shallow soil loosening, conservation agriculture, and then is the mean. And everything on the right side, it means improves, increased earthworms. So the less disturbance to the soil, the more earthworms you have. So of course, conventional tillage is really bad for worms. And we also discovered that it will take 10 years for those populations to recover. So the next question is, if to be good indicators, they have to be good indicators of processes. So one of the processes, of course, are the physical processes. Aggregation, we tend to look at microbes and all that produce aggregates, but also worms produce aggregates. And in collaboration with a group of Portuguese from Coimbra, we did this sampling of earthworms in different agroforest systems, and we found every time we increase earthworms, we also increase aggregate stability. So, but earthworms also play a key role in chemical processes, and that has been studied quite a lot. But the thing you always have to remember, not all worms are the same. We have, well, there's a big discussion whether it's a functional classification or not. I won't call it classification. We have three groups of worms, the ones that live on the surface, which are the epijakes. Then we have the endojakes that live in the mineral layers, and then we have the anesics that go up and down and feed and cast on the surface. They all produce burrows, which is a physical structure. They all produce casts, but some of them, they produce the casts in the surface, and others just covered all the burrows with casts. So that what happens is what I consider hotspots of mineralization and retention. And the epijakes and the anesics, they also have the lovely characteristics that produce calcium carbonate granules that they're released into the surface, and they are CO2 traps. So there's another way of how worms also can sequester CO2. So we need to know whether in our system we have the three groups, only one, two, or none of them. So the other important thing I think we need to consider with indicators is which are the direct and the indirect effects. For me, direct effects are, of course, the burrowing, the feeding, the breeding, the casting they produce, and also another thing that nobody considers is when they die, they also release nutrients that also goes into processes. But of course, we have also indirect. Worms interact with microorganisms, the casts attract microorganisms. They also produce plant growth regulators, so they increase plant productivity, and also the plants release nutrients that the worms can and later they can feed on. So there's all these interactions. So the other question is there any other good biological indicators in agricultural soils? Well, with Carmen, who is my postdoc at the moment in my lab, we are doing this work. We apply the David Wardell Index, which compares conventional treatments against organic or sustainable. And everything above the line means that are stimulated by the conventional treatment. Below, they are inhibited. So, of course, if you are in Gitrate and Asinfilan, you are also could be an indicator that is a very aggressive tillage. So here with Ladislav, we consider that when you look for indicators, you have to know whether it's a pristine system, which is a natural state, or what we call a cumulative regime, where positive impacts and soil biota are doing the job for you, and they are producing the good ecosystem services. But if you are stressing the system with agriculture, with pollution, then the soil biota don't have opportunities, and then unless you add external inputs, you won't be able to do what the soil biota do. And to recover that only based on natural processes, it will take a long time. So I think we should better target our organisms depending on which ecosystem type we are considering. And here in this graph, what I want to show is we have different players depending on the ecosystems, but also they have activity points. It's like, for me, like switches. So, for example, here, in the winter, most of the time in the boreal forest, nothing happens. It's a narrow window when there is all this activity in the soil and in the plants. Then you go to the temperate regions and tropical regions, you have a huge diversity of players. They move down and they can extend their activities down in the profile. So this is they can affect soil processes deeper in the soil. And then you go to the arid regions, you have the opposite. It's only, again, another different windows when most activity take place. So when I go back to the soil monitoring law, that was my reaction. So the countries have to choose the indicators. I think it's a mistake to give so much flexibility. But also some countries, as it was pointed in the lunch before, need funding because the compulsory indicator is metabarcoding for no reason. I didn't see any reason. Except for, is in Lucas. Is the cheapest? No. Is the easiest? No. You really need not only to be able to extract the DNA, you have to sequence the DNA, you need bioinformatics to align all these sequences, and then you have to interpret all those sequences. Can all the member states do that? And when we look where the funding goes for is only to the big animals, very few to the invertebrates. And then when we look at the protection regulations, they are all scattered and also mostly on birds, vertebrates, mammals. I mean, there is a soil biota down there that says others, very, very fine. So, and most of the things are based on above ground biodiversity. So, another question was, are actually the current protection areas like national parks or the Nature 2000 network good for protecting soil biodiversity? So in collaboration with Romy says, we look at the earthworms and we predicted that most of the worms with climate change will shift in the distributions, and more or less 80%, more than 80% of them were not well protected by those figures. So of course, the Habitats Directive is very good for protecting habitats and protecting plants, but that doesn't mean that we have to assume that it's also protecting soil biodiversity, because it's not happening. So how we can protect soil biodiversity? Well, we need to talk to the lawyers. We need to talk to the policymakers. And the problem is we talk in different languages. So one of the interesting exercises for me was to collaborate with this bunch of layers from Wageningen University, and we start to see how we consider soil. And in the left is how they consider soil, and on the right is how we consider soil. So soil is something that is, we need a flat rate to regulate, whereas we think it's a complex system. They all think that it has to be one indicator or something, and we say, as I said, well, it depends. How many? For them, soils is a commodity, whereas for us is something that is not renewable. It's very difficult to regulate because it's very difficult to control what is doing what to the soil and whether it's good or bad. So we need to protect soil and also soil biodiversity. We need to strike this connection. So with Julia, who is also here somewhere in the audience, I hope, she did this review. We find out that at the European level, all the regulations were scattered in different frameworks and documents, but there were only very few that explicitly mention soil biodiversity. And many of them were saying, well, implement sustainable practices, assuming that that will increase soil biodiversity. And I believe we don't have to assume. We need stronger measures to make sure that we actually protect soil biodiversity. So the solution is to talk, and it has to be top-down, but also bottom-up. So, of course, there has to be a policy body that tells us what is the good thing and how it should be done, but it should be fed by the bottom up, by scientific knowledge, science basis, and also by the people that actually are using the land or using the soil in some ways. So now come to the project, European project and coordinator, because that gave us the opportunity to tackle many of these points. The soil mission is a €1 billion program that produced funding for Europe for different groups, and this is the project I coordinate, which is Soil Biodiversity for Ecosystem Services. These are all the partners, and that is with the aim to implement that soil directive I was meaning before. So we are using lots of indicators. We are also using traditional methods, morphology, but also molecular. We're going beyond just using ventabar coding, like Lucas, and also just only using morphology, like in soil-borne. So we're validating that identification, that sequencing with morphological, and we're also applying different techniques for community structure, the DNA. We have sampled a bunch of sites across different countries, different climatic regions, and we consider 3 different land use, and in each one of them 3 land use intensities. According to the directive, we're also separating the forest, the organic layer from the mineral layer, and we are getting now the first results. So we are comparing pedoclimatic regions, land uses, countries, and we're also doing temporal sampling. So watch this space because there will be interesting results coming out. And of course, like everybody else, we produce our handbook of methods, which we think is the best. And every other project produced the handbook of methods. But we also heard in several talks, there's also Lucas methods, and there are also soil-borne methods, and we just now got accepted the soil-borne food web methods. I like all these words that appear on the top. We have to go for the standardized methods, harmonized. I like this word harmonized because it looks like a symphony, a melody, but to me it's actually a cacophony because everybody's fighting who is the best methods. And I think it's a completely waste of time to do that because we will never agree. So I think we better think ways of integrating the new data we're producing and the old data into large initiatives, like some of the ones they are present here today. I mean, Solbot, NETSOB, but also the European Soil Observatory. And then we can respond to what the policymakers need and how we integrate the data. Well, that is the big question. In fact, I had a chat with a taxonomist, and he said to me, I don't understand ecology papers, because I am a taxonomist. I describe a species, and this species, if it's found in another place, it will be the same species. There's always the same characteristics. But you ecologists, you do experiments in pots, in different systems, with these conditions, to this depth, sample, in different occasions. How do you compare all these? So that is a good question. How to make this data comparable? Well, the only way, in my view, is to do a very good data structure, and is to record and to annotate everything we do. So here's an example of what we're doing in this European project, but it could happen to any project. So we have a project, and we have a set of methods. We have different ecosystem types that we define what is a forest, what is a grassland. Then we have methods to collect the samples, how we describe the sample, how we process that sample, how we prepare them. So then how we storage the animals or whatever, or the soil, and how we analyze, we analyze which properties. And then if we record all that for every single step, we will be able to even go in more detail and include all our metadata, specify which units are using for each property, and then if we find all this, we can trace back, we can do a tracking back of our properties. So this is what we are doing in this project. We call it Digital Data Warehouse for Software Ease, and what we're trying to sell to the European Commission is your job is to combine the nice information that all these European projects are produced, that everybody can use and everybody can integrate. But it's only when I know how that data has been measured. Let's say, for example, the EU wants to know which soils have high content of aluminium that has been measured with this specific ICPS or whatever. Because it's being tracked, the system can select those samples that have been obtained by this method. It's not good if I take a pH with potassium or with calcium chloride, and then I say, well, in peatlands you can have this range of pH. So in this system, of course, for the moment, because we are trying is an internal system, but it will be open. You can upload your data. Each data row is being tracked. It has a unique ID, but you can also drop your Excel file, and the system will start mapping your columns. So if I have pH, it will suggest, well, is this pH in water or is it in calcium chloride or what it is? So it will be, and that's the only way we can make data sharing, what is called data interoperability. Is the ability of being able to share data, to integrate data comes from different sources. So my message is, instead of looking and trying to find which is the best method, I think we should be able to make more efforts to integrate data. So what are my conclusions? I want to keep like a kid and asking always over and over, why? So measuring everything everywhere, like some of these directives, why? Should we now focus on quality instead of quantity? And my suggestion will be, instead of, well, all the peatlands in Europe, they should measure enkitrace, for example. And maybe in agricultural soils, you could measure nematodes and maybe earthworms. So instead of going for a flat rate, like a telephone company, that you can use and it doesn't increase the price, I suggest different systems, different contexts, different climates, a set of indicators that it will inform about that system, instead of having the same indicator for everybody. So the other thing is to have, of course, cost-effective indicators. But what the big, large programs want is something that is easy to measure. Well, counting worms is cheap. You just make a hole, start collecting the worms, and you count it. And that biomass and abundance is already very informative. Enkitrace, well, you can collect a soil core and put the sample under the lamp in a funnel with water, and it's cheap. Anybody can do that. So I don't think, I challenge them to argue that they are not cost effective. And then the next thing is what I said lastly, I mean, it has to be very good in the way we report the data. It has to be well structured to describe how we collect those organisms, but also the metadata is very important. That is often missing from these large monitoring programs. And here the experts divide what is hard metadata from soft metadata. So hard metadata is the information relevant to the property you measure and assess, where soft metadata is the reason of the experiment, the rationale, whether it's in the field, in the pots, in incubations. So you have all the information regarding that data, that number. And then, of course, I think for a soil monitoring program, ideally, instead of giving so much flexibility, I will ask for a minimum number of indicators that all the members should measure in the different ecosystems. Instead of one country measures 5, another 20, another 1. So I think it's better if we agree on that. So that's it. Thank you very much for your attention.